Okay, I think we'll try and get started. Again, welcome everybody. Thank you for taking time of your Saturday, your important and valuable family time to be with us today. You know, at the foundation, we've always had that feeling that it takes a village. It takes a village to support and or help find treatment for, a cause. And so we are really thankful that all of you can join us today. These webinars are really meant to show how you can help and how you can contribute to the cause at hand, influencing research in a positive way so that we can actually move treatment along to be more effective and earlier on in the process. You've probably all seen different documentaries and movies about how patient organizations have helped improve the knowledge that researchers have and the path that they follow, and also bring greater passion level to the researchers when they start meeting people that they're serving. All of those things serve to have a positive impact. I just want to assure all of you that we are a mature foundation with all the elements to be successful. And we're going to go over a little bit more about that today. All right, today's agenda, we're going to go into a little bit more detail about what it means to be a community research partner. So we're going to have some overview on that. A few more details and reminders about the SciFam conference that we'll be hosting, both in person in Nashville and virtually. And then really wanted to talk about today the importance of research resources. The researchers really want a rich platform to work from. And when they have things like well-developed cell repositories, places where actual human tissue can be harvested and grown for research to actually mimic the human form and be tested, is really an important fact. So we're going to go over a little bit of details today about what resources we have set up there. The CMDIR, I'm sorry, can you go back one? Thanks, Kate. The CMDIR is our registry. So we're going to review a little bit about our registry details today. If we can't access and find people to be involved in research studies, that hugely impacts our ability to actually do discovery work. So our goal this year, and you'll see it on a slide later, is to actually double the number of people that are registered in the CMDIR. Again, the Congenital Muscle Disease International Registry. The Tissue Repository, the CMDTR, is we're going to hear a little bit more detail today about the cases that we have, but this is actual human tissues and cells that are stored so that researchers can request the different valuable options for their work. And then there's a little bit of a difference between that and the Congenital Muscle Condition Biorepository. And Stacy will do a nice job talking a little bit about those. And then we have the good fortune today of having Dr. Mike Lawler with us. He's going to review the importance of human tissue resources, and that's going to be an important part of our talk today. Looks like it skipped a slide, Kate. Can we go back? Yeah, great. Okay, so then this is, again, great thanks to our team that are putting in tireless hours of work. I'll let you just peruse the names here. We also have a stand-in for Terry Saluki, who's on maternity leave. Michelle is helping us with our grant writing in the meantime, but just really grateful to have this team with us, every day at the table, willing to serve the community. For those of you who didn't see our timeline at the last webinar, I really want to put this out there and really just keep coming back to it with clarity, is that by the end of this year, we hope to understand the effects of gene replacement, utilizing CRISPR on NM animal models. We hope to understand what medicines cause Nebulin zebrafish muscles to be more active, stronger. We want to increase, like I said, the registry participation by at least 100%. By next year, our hope is that we'll have 2 companies actually having tissues in their labs and testing their compounds that have already been created and deemed safe on these tissues to see what would help those with NM. In a few years, we hope to complete the natural history study to actually have all the data and details we need and the FDA to be able to approve a clinical trial, a clinical study. We also help to have a better understanding of the pathophysiology of the rarest cases of NM and what therapeutic modality would be feasible for those cases. In 2025, we want to identify the best NM treatments to start preclinical studies and also establish interaction with the FDA to agree on the protocols and pathway toward regulatory approval of treatments for NM. And then by 2026, which is only 4 years from now, launch a clinical trial with an industry partner on supportive treatment. And then finalize the discovery of genetic repair for a curative mechanism. It's only 4 years away, and those are our ambitious goals. We can only do it with your help. I'm going to take a minute just to introduce Sarah Foy. She's our outreach coordinator for our PCORE project, and she can review all the details that go along with our commitment to this award.
Hello, and thank you, Mark. So what is PCORE, which really is the patient-centered outcomes research? And the project that I'm helping with for a foundation building strength is for the PCORE award. And patient-centered research involves active partnership, which Mark touched on. And that is what today is about, and also our conference and this whole project. So the aim of the Foundation Building Strength PCORE Award is to empower this community to be partners in driving forward meaningful research. And it also is to create a lasting infrastructure to support research initiatives into the future. So thank you very much for being our partner and for listening to this webinar today. And I'm really looking forward to hearing from our next speakers, which include Stacy Cassette. So I'm going to go ahead and turn this over to Stacy.
Hello, everyone. Thank you so much for joining us today. I'd like to start by just doing a little bit of review of the CRP role and expectations over the next 18 months. As all of you know, I've had a number of email correspondences with you about some of this stuff, but there have been some questions that have come up. So hopefully we can review what those things are and get your questions answered with this today. So basically there are some attendance expectations, and that includes the webinars, such as the one you're attending today. We do hope that you'll be able to attend at least 5 and up to 9 of the webinars that will be hosted over the next 12 to 18 months. These will all be hosted via Zoom, so you can attend just from the comfort of your own home. They'll generally be hosted on a Saturday, like today, and I think this time of day seems to work well for people. Excuse me. We also have family advisory board meetings that will be hosted, and you as a community research partner are automatically a part of that advisory board. The first one is going to be scheduled July 1st at 3:30 Central Time. This is going to be during the SciFam conference. So those people who are attending in person will attend this advisory board meeting in person. And those of you who are registering virtually for the conference will attend through the conference portal. And then also the SciFam conference, as I just mentioned, there are both virtual and in-person options. The conference for families is going to go on Saturday and Sunday, July 2nd and 3rd. There'll be sessions that you can attend just based on your preferences. And then also there are plans for a virtual NM convention sometime next year. And what we're really looking for from you is your perspective. We hope that you'll be engaged during the webinars, the meetings, conferences, and so forth. We hope that you'll complete the surveys, which will be a great way for us to get your perspective. And also just provide your input on the various NM initiatives that AFBS has going on. And finally, we do have plans to develop a resource or welcome kit, and we'd like to have your perspective on that as well. And finally, as you know, we're offering a stipend of $300 for those CRPs who can fulfill the minimal expectations for the role. A number of you have responded to me that you would like to claim it or that you are not interested in it. and either way is perfectly fine. We are planning to send out those checks probably in early fall. Next slide, please. And also, I just wanted everyone to be aware that teenagers are welcome to join this initiative. Anyone aged 13 to 17 will require parental permission, and we do have a parental consent form. So if you have a teenager that's affected with Nemaline myopathy that might be interested in this, please contact me. I'd like to touch just a little bit on registering for the conference. SciFam 2022 is our Community Scientific and Family Conference, which will be held in Nashville, Tennessee this year. June 30th and July 1st will be for our scientists, and July 1st through the 3rd will be the patient family gathering. As I mentioned, July 1st, we have an afternoon meeting at 3:30, and then there will be a welcome for those who are attending in person at 4:30. And the sessions go on Saturday and Sunday. Everyone does need to register to gain access to the sessions, the meetings, discussion groups, and so forth. The virtual registration, which is normally $125, will be just $50 after the AFBS exclusive discount. In-person registration, also normally $125, will be $0, free after the AFBS discount. And those who do choose to attend in person may be offered a $600 Visa gift card to help offset your travel costs. And that gift card will be available on site, either at check-in or during Friday's meeting. And here is where you would go to register. It's scifam.info. The website has a lot of great information. It has agendas, and of course, the buttons that you would press to register either virtually or in person. If you would like to see an instructional video, we do have those available, which will walk you through how to register either virtually or in person. And those links are going to be provided to you. Next, I would like to get into some of our research resources. So these are a very important part of our infrastructure. This is an important way that families are able to contribute to advancing research forward even faster. The research resources that we have in place include the Human Genetic Cell Repository at Coriell, the Congenital Muscle Disease International Registry, the Congenital Muscle Disease Tissue Repository at MCW, and the Congenital Muscle Condition Biorepository at MCW. Now, these programs all work together and each contributes in unique ways to the overall goal of helping to improve our understanding and find treatments for congenital muscle diseases. I act as a central coordinator for the programs, sort of linking them all together. And this involves making direct referrals, data management, providing information and outreach services. And since each program is run from a different location, each must follow the rules of the facility that oversees the program. And this is why there are separate forms to complete for enrollment in each program, which are mandated by the oversight committees of each facility. Now, just to overview what each of these is all about, the cell repository is the donation site for blood and skin samples to be made into cell colonies, which are marketed to scientists. The registry is the online patient family registry for storing information about our participants. The tissue repository is the biorepository specifically for congenital muscle diseases to centralize and distribute tissue samples to scientists. We also have an associated postmortem program. One important priority that all of these programs have in common is protecting your privacy. To do so, a code number is established for each case or sample. We remove personally identifying information, and we follow strict guidelines that forbid us from sharing any identifying information. Okay, let's get just into a little bit of detail about the cell repository at Coriell. Again, this is just going to be an overview. So I'm going to be really brief about everything. This program was established in 1972, a lot of longevity there, in New Jersey, and they do store disease and healthy specimens. The program is compliant with all national and international laws and is IRB approved. This program catalogs transformed lymphoblasts, fibroblasts, induced pluripotent stem cells, and DNA. And in the last check that I did of their catalog, they had 154 patient cases identified as having a congenital muscle disease, plus unaffected family members. The cell repository can accept specimens of blood or skin biopsies from patients and immediate biological relatives, such as parents, children, or siblings. And this is for a one-time donation that is then made into cell lines for research use, using the patient family's exact genetic variant, which is very powerful. If you would like to read more about this program, just go to coriel.org. And here I have shown a screenshot of what the affected patient family page looks like. Okay, next, I'd like to get into a little bit about the registry. The patient registry was launched in 2009, and currently we have 250 identified as Nemaline myopathy enrolled from all across the world. This program is compliant with all national and international laws and is IRB approved. This is a self-completion format using online forms and requires you, the user, to provide updates as needed. The purpose of the CMDIR is to raise awareness and standards of care, assist with enrollment for clinical trials, support our researchers with goals to find treatments and improve care. And it also can provide de-identified data to our scientists. As an example of some of the data that we do provide: age at the time of sample collection, genetic diagnosis, maximal motor function, breathing status, and the level of heart or brain involvement. I think you might have skipped one slide. There we go. So if you are interested in registering or still need to reclaim your profile on the new platform, just go to cmdir.org. If you have never registered before, what you do is click the orange button, which says register. If you have registered in the past, but have not gone in to log into your profile within the last year, then you will need to do what's called reclaim your profile. And this is because the registry was moved to a new platform and went through an extensive upgrade. So what you'll have to do in that case is click on Forgot Password, and that will send an email to you with a link that will help you reestablish your password on the new platform, and then you can get into your profile and update everything that way. If you have any problems with this, please email info@cmdir.org to get help. We do have plans, and AFBS is intending to hire a registry specialist, but until that person is hired, please use the info@cmdir.org to get assistance with your profile. Okay, next let's talk a little bit about the tissue repository. This program was launched in 2013 at the Medical College of Wisconsin in the Lawler Laboratory. We have 83 identified with Nemaline myopathy enrolled, and 47 of them have donated tissue samples. This program is compliant with all national and international laws and is IRB approved. Some of the possibilities for donation include leftover tissue from a previous muscle biopsy that you've had. And we can bring in tissue from biopsies that are very old. I can think of a couple of situations in which the muscle biopsy had been done 16, 17 years ago, and the facility did save the tissue, and we were able to transfer that to our biorepository for research use. Also think about surgical procedures you have coming up, because oftentimes this is a great opportunity for collecting a small sample of muscle. A lot of our patients are having surgeries, contracture release, scoliosis correction, and these all might be opportunities for a sample collection. And then also samples may be collected during postmortem exams. The basic process is you consent for the program with me, and then I work with you to identify a donation possibility based on those examples mentioned above. I would consult directly with your clinicians and then make arrangements to have the tissue transferred. The CMC Biorepository is an affiliated program, and it's a place to store organ and tissue samples that were collected during pathological exams after death that can be used for research to help understand more about the muscle disorder and to help develop treatments. Basically, our biorepository programs provide a service to affected individuals and families who are interested in supporting medical research and treatment options through tissue donation. The tissue is then made available to scientists who study that specific muscle disease. If you would like to learn more about this program, please send me an email, stacy.cassette@cmdir.org. And if you'd like to learn more about any of our tissue banking, you can find that information at mcw.edu/cmdtr. We have a lot of great information on the website, including frequently asked questions and the guidebook for families and patients called The Care of Congenital Myopathy can be downloaded for free from this website. So why would you want to participate in the CMD Tissue Repository? Well, this is a centralized human tissue bank, which can accelerate research to finding effective treatments faster. And without your participation, these samples are just simply not available for research. Your or your family's exact genetic subtype could be available for these studies, and that is so powerful. This provides greater insight to our scientists into how the disease affects patients, how the condition changes, and how cells might respond to treatment strategies. And with this, I will segue into the introduction of our guest speaker today. We are very honored to have Dr. Michael Lawler with us today. Dr. Lawler serves as director of the CMD Tissue Repository and Congenital Muscle Condition Biorepository. He is a professor of pathology, neurology, biomedical engineering, physiology, and cell biology, neurobiology, and anatomy, plus director of 2 clinical laboratories focused on the diagnosis of muscle and nerve biopsies at the Medical College of Wisconsin. He is also CEO, founder, and owner of Diverge Translational Science Laboratory, a commercial research laboratory that facilitates the translation of treatment research from preclinical through the clinical stage. So thank you very much for joining us, and I will turn it over to Dr. Lawler.
Thank you, Stacy, and thank you to the organizers for inviting me to speak today. Let's see. Do I just ask for the next slide? I don't. Yeah. Okay. So I have a whole bunch of disclosures. As Stacy mentioned, I'm very active in working with companies and transitioning or developing treatments for muscle disorders and other metabolic disorders, and that's puts me in a good position to understand what's out there, and then ideally, in the case of Nemelin myopathy, also be able to talk to these companies about Nemelin myopathy and do the best that I can to get Nemelin myopathy onto their radar as a possible pipeline option. Next slide, please. So I'm just going to give a quick talk today about sort of what the I think we need to go back one. No, maybe back 2. Yeah, but back one more. Sorry. Yeah, thank you. I'm just going to give a quick talk today about how the tissue repository gets used. And so the idea here is that patients like yourselves donate to the tissue repository. They consent with Stacy. The tissue gets sent to us at MCW. We store it until somebody has a use for it, and then we distribute it. And in my own research lab, we've used some of the tissue. There's also a number of projects that have been outside of my research laboratory that we that we've sent out tissue to, and they've been able to use it without our further involvement. And so what I'm going to talk about today is kind of the general uses, but also some examples of ways that we've collaborated with people around the world to use donated tissue to make a variety of scientific discoveries and steps forward in understanding the diseases and developing treatments. So when you think about how donated tissue can be used, it can be used to answer a number of questions that really are not necessarily all answerable using animal tissue. So in the most basic form, you can look at tissue in terms of its structural problems. You can also use old tissue in some respects to look at how the function is abnormal. So there are scientists that have the capacity to take, for instance, a frozen piece of tissue and treat it in a certain way that they can look at how well the muscle contracts. It's not necessarily always as useful as fresh tissue, but there's still a great deal that can be learned. And, with respect to looking at structural problems, usually there's no difference between looking at a sample that's 5 years old versus 5 days old if it's been treated appropriately in terms of its handling. One important thing to think about in terms of human tissue really is that when you think about treatment development, understanding diseases and all of that, people really rely a lot on animal models to do that, but the animal models are all essentially genetically identical. And so there's a lot of unanswered questions in relation to how much variation can happen between patients. And myopathy is a really good example of that. There's a lot of people that have mutations, for instance, in ACTA1, but the symptomatic severity that you see with ACTA1 myopathy is all over the place. And yet there's only a few animal models of ACTA1 myopathy that kind of addresses that. And so when you have the access to human tissue specimens, you have the opportunity to really answer your questions across a broader range of ages, genetic subtypes, things like that, what's happening in different muscles. And that's, as we'll talk about, important when it comes to transitioning to a human clinical trial, because you need to be able to have appropriate expectations of what's going to happen in humans in comparison to what happened in the animals themselves. More broadly, right, we use the tissue to answer questions about what's going on in the diseases, also identifying treatment targets and useful treatment options, ideally also to be able to predict outcomes. And myopathy is another good example of that, where at the time that you, at the time that you may be diagnosed with myopathy, they may not be able to tell you what the clinical course of it's going to be. Ideally, human tissue studies will help answer questions to be better informed at the time of diagnosis and while you're, while you're being cared for. I'm going to move forward now because the remaining points are actually on the next couple slides. I'm just going to move through a couple of examples of how we've used the donated tissue. So one example here is primary disease characterization, and myopathy is a good example of that. So here's a paper that we did in collaboration with Shamindra Konersman at UCSD. The idea here is we had myopathy patients for which there was no genetic mutation known. And we worked with Shamindra and some geneticists at UCSD. They were able to do a detailed genetic analysis of the tissue, and they found some unusual mutations in troponin T1 that wouldn't have been found on routine clinical sequencing. And so by donating the tissue, we were actually able to push forward a research program that allowed for the identification of new genes associated with, or new mutations associated with myopathy, and had some ability to correlate what was going on with what was present structurally. Next slide, please. Here's a project that we've done in collaboration with the CMDIR and with Prothelia, which is a company that was focused on LAMA2 congenital muscular dystrophy. And the idea here was actually to transition a bunch of anecdotal knowledge that had been accumulated over the years about what happens in LAMA2 congenital muscular dystrophy, and to produce a presentation and a paper about some key aspects of the disease, because the idea here is that when you propose a trial, for instance, to the FDA, you're going to need to substantiate, for instance, why you would want to treat young patients at an early stage of the trial. Because, for instance, the severity of patients and the pathology occurring in the older patients is going to make it less effective to see treatment efficacy. And so in this study, we had a number of patients from the CMDTR and a number of patients that had been donated tissue to a biorepository in Iowa. We were able to get about 60 patients worth of samples and look at how their pathology looked according to a couple of different markers, and then correlate that with things like their age and their sex, things like that. And so the idea here is to get a better understanding of how a couple of key disease phenotypes change with age, and also to do a very thorough characterization of what is going on in these samples for a fairly large cohort. And so just what I'm showing here in this picture is that you can see that the H&E between the congenital muscular dystrophy patient looks a great deal different from control. The muscle fibers there are very small, and there's a lot of blue inflammatory cells there. You can see there, as expected, that there's not any LAMA2 at the edges of the cells. So that's why there's no green in that top middle picture. But one thing that was a bit surprising to us is that some of the other laminins, in this case LAMA4, were being upregulated at the membrane in some of these patients, but not all. And so part of this project is looking at these broad range of phenotypes so that we understand what are good things to track and what might be good things to follow up on. Next slide, please. A similar thing was done when we were working in the Xlinked myotubular myopathy field. And this was a run up to the Aspero clinical trial for gene therapy in myotubular myopathy. And so we worked with Audentes Therapeutics and a number of experts in the field to really categorize the pathology that was occurring in the mouse models, in the canine model of myotubular myopathy, and in humans, and really highlight how things were similar and different between these. And the idea behind this publication was actually to educate people like the FDA to understand that when they look at a mouse paper about myotubular myopathy and they see that certain things were done and they weren't done, for instance, in the human clinical trial, the reason that a lot of that happened was that the pathology was different, or that the tools that were working well in the mouse to detect something didn't work well in human tissue. And so, there's a lot of utility in using these banked human tissue samples to show how, set realistic expectations for what's going to be possible in demonstrating treatment efficacy in human patients. Next slide, please. Logistically, this also can be very useful. One of the things in that Aspero clinical trial that needed to be decided was how to do muscle biopsies. And we needed to do 3 muscle biopsies, and the muscles were very small. And so we didn't want to keep going back to the same one because that would add a significant burden to one limb, for instance, and also make it less likely that you were going to get a good sample. And so we were able to use some of the autopsy samples that we had collected over the years to look at the differences between the gastroc muscle and the quadriceps muscle, and then really decided that they looked essentially equivalent. And so if we needed 3 biopsies, which is what we did, it was acceptable to take one from the left gastroc, one from the right gastroc, and then one from one of the 2 quadriceps muscles, and that we would expect to see the same things. And so it's important when you're planning these things to have some sort of proof of your expectations rather than assumptions. And so things like these tissue repositories allowed us to make informed decisions about how to design the trial and the tissue collection there. Next slide, please. The other really unfortunate use for the tissue repository is looking into patient safety outcomes. And so, as many of you likely know, there have been 4 patient deaths in the Aspero trial. And we've been able to use tissue repository samples to help understand what was going on there. And so, these 4 patients had undergone gene therapy, and then a couple months later, they died of complications of liver failure. And in looking at the samples from these patients, the autopsy samples from these patients, we realized that they were undergoing a specific subtype of progressive liver failure, and that it had the potential to have a component that was pre-existing, because we were only seeing this in this MTM trial. We weren't seeing it in other gene therapy trials that had similar doses and similar approaches and things like that. And so actually what we were able to do is we were able to go back to some banked autopsy samples from myotubular myopathy patients and see that in a subtype of these cases, we were seeing some very mild liver disease that was that looked like essentially the mildest form of what got worsened by the treatment. And so there's a predisposition in myotubular myopathy patients to having this kind of outcome that wasn't recognized and wasn't known. It wasn't in the animal models, and it wasn't necessarily in all of the patients. But we were able to track that down a little bit. Conversely, some of the findings that we were seeing on the autopsies, like the downregulation of this BSEP protein, that's a key bile transporter, that wasn't a problem in the untreated myotubular myopathy patients. And so in using this banked tissue, we've been able to find a few associations that give us a clue about how these fatal outcomes developed. And that's really the first step to figuring out how to avoid them in the future. Next slide. And so overall, in terms of the CMDTR usage, we've been consulted by 32 research teams about their needs. We've distributed it 17 times across 59 patient samples. You know, I talked about a few of the outcomes related to work that I've participated in. We actually don't have follow-up. We can't request follow-up information on how outside labs have used the tissue. It's just an element of how we're regulated. So, you know, if we had that information, then we'd need to report it back in our IRB, and then there'd be a lot more regulatory burden associated with it. But, you know, we do know that at least the outcomes that we've gotten from the tissue have been great, and several of our collaborators have also benefited greatly from it. With respect to, you know, when people ask for the tissue and it doesn't result in a distribution, I'd say the most common thing is that they request either tissue that we don't have, or they produce a request for the tissue, and we say, okay, you need to do this minimal amount to demonstrate that you have ethical oversight for the use of human tissue, which is about a couple hours of work to submit to their IRB and then wait. You know, there's just a certain subpopulation of investigators that never go through that. And actually, that's kind of a good litmus test for if somebody was actually serious about using the tissue. If they're not willing to do a little bit of regulatory work to get permission to use the tissue, then they probably weren't going to usefully use the tissue anyway. And that, you know, that allows us to have a little bit of a gatekeeping function as well. Next slide. And then just to kind of finish up, you know, in terms of looking forward, I think there's a lot of therapeutic development going on in muscle disease. You see that you know, AFBS has a very, you know, aggressive timeline towards supporting the development of treatments and getting to clinical trial readiness. This kind of resource, having this kind of resource available to set up the logistics of trials and to understand the types of questions that you can't interrogate animal tissue with, is really important in making that transition. You know, it's really important to have access to human tissue to donate, to understand genetic disorders for a lot of the reasons that I've talked about, because it's just, the animal models and cell culture models are great, but they have limitations. And increasingly, we're at a point in therapeutic development where the differences between humans and animals are really a big limiting factor. The other thing is that, you know, while it looks like we have a lot of samples, those samples get used. And so we always need to enroll as many people and as many samples as we can to keep up with demand. I think the demand to use these samples is going to go up steadily over the next couple of years as well, as people see these samples being used, you know, more publicly, and it's apparent that they exist and that they can answer the key questions in therapeutic development. And, you know, as in a couple of the cases that I pointed out here, we don't even necessarily understand ahead of time what we're going to use these for. Like the myotubular myopathy liver problem was not on any of our radar. And yet that's been one of the more important uses of the tissue repository over the last few years. You know, that's why you bank what's available and you make sure that you have it in a good state in case you really need to be able to investigate something on a short time scale when patient outcomes are really on the line. So I'll finish there. This is a picture of my wonderful lab group with Stacy photoshopped in there in a very professional looking way. And it's been a wonderful journey with the people at AFBS and the other organizations that support the tissue repository. And I am grateful for their continued support and involvement in this.
Dr. Lawler, thank you so much. That was great. I'd love to open it up to any questions at all. Stacy, I'm not sure if you saw Joanna's question in the Q&A. There was a question that says, can you send tissue from Europe to the United States? Here we go.
Hi, yes, I did just see that now. So we had a little bit of a complication with our oversight committee in response to some new laws that were passed in Europe in 2018. So in 2019, our oversight committee thought that bringing in tissue from the European Union was going to involve some risks that they didn't want to take on. And so as a result of that, we stopped bringing in tissue from the European Union as of 2019. But what we're going to do, Johanna, to answer your specific question, is bring that tissue in through the BEGS lab. So it is still possible to get that tissue over here and make it available to our scientists. We're just going to do it through his lab instead, where they don't have the same type of burden associated with bringing in samples from the European Union that we have now.
Thanks, Stacy. There's a new question also. Tough to see who it was from. It just says admin, but is it possible my son's sample from 2008 is still around or used up?
Yes, it's very possible that it is still around. And I just want to explain a little bit more about how this works. Really, the patient families only need to consent, which involves looking at a form that is about 4 pages long, just explains the program. You sign it, you date it, I sign it, Dr. Lawler signs it. And that's really the extent of the burden that's put onto families. At that point, I simply find out where the muscle biopsy was done. Or in the case of a surgery, I find out where the surgery is going to take place and who the surgeon is. And then you're off the hook as a patient or family. I take on the next steps myself. So I make direct contact with your clinicians. I find out if the tissue is available. I find out if the surgery you have coming up might be an opportunity for tissue collection. And then I make all the arrangements with your clinicians directly. So you generally don't hear back from me until after all of the arrangements have been made. And then I send an email to families just letting them know as an update, the tissue has been transferred, it arrived safely. Thank you very much for your contribution.
I want to thank Erin for that suggestion and that extra review, because she actually, that does take a lot of burden off of the families when it's being done for you. I see Cindy asked a question. I guess how is tissue managed when it is in the tissue repository? How is it managed when a researcher makes requests? And are they not required when they have taken tissue to disclose the outcomes of that research? I don't know, Stacy or Mike, if you want to handle that one.
I can start, and then Mike, you can add in, to fill the gaps. So basically, when a researcher is interested in tissue, they fill out a form, and it's a tissue request form. And on that, they specify their intended use for the tissue, how much tissue they anticipate needing, which genetic subtypes they're interested in, a lot of detail like that. Then that request form goes through Dr. Lawler. He determines if that is a good use of the resources that we have. And if so, it does get approved. When the tissue is distributed to a scientist, we do not require that they report back to us on any of their results from their studies. There are some situations in which Dr. Lawler is a collaborator on studies, and in that case, he would have information on the outcomes. But in other cases, we just simply don't know what the outcome was of their study.
Yeah, so I think that's an excellent, it's an excellent question. It's an excellent response on Stacy's part. So really the thing we ran into was that if we sought results, then we would be on the hook to report back to the regulators within our IRB exactly what was going on in every sample that we had distributed. And so we were given the choice that we either had to chase down people and get them to update us on every donation, and be required to do that by our compliance at MCW, or that we could distribute it and then have as a condition of it, which is on the consent form, saying, listen, if we distribute this out, we may not know what happens to it. And that's just going to kind of need to be okay. When it was made clear to us that it was going to be a significant problem if we went out, asked for updates, and then people didn't update, so that we would get in trouble, but that they wouldn't, then it was just decided that it wasn't feasible for us to require people to do that, because it would put too much requirement on us to tell our IRB what was going on. So we kind of had to back off from that. Yeah, Stacy said, by and large, about, I don't know, what would you think? About 80% of what we distribute is something where, we're actively involved in the research anyway. We distribute it and it's a close collaborator, and we kind of know, what ends up happening to it. But we also didn't want to restrict it to, something that would make it look like my personal tissue bank, and that I was only giving it out to my friends, which is a concern, right? Because I don't have that many friends. So, so in a nutshell, that was it. It was the kind of thing that on the basic level we wanted to be able to do. And then when we said, well, we'd like to be able to do this, they said, well, then we're going to require you to report back to us all this stuff. Mm-hmm. It just wasn't feasible.
This is Patty Mitchell. I'm sorry for jumping in. I do want to make it clear that the foundation does sponsor studies, and we require reports back on those studies. And so it may be being used for another study outside of our foundation. I would guess anybody spending money on a study would require a report back on the results. So I don't want you to think from this conversation that there's no reporting back. There is. It's just that the tissue bank themselves isn't getting the reporting back. The people actually funding the studies are. So I just wanted to make that clear. Then I'll come off.
Yeah, that's a great distinction.
And I also want to add that usually these are research labs, and they grow and they keep going because they publish the data. So usually you will see the report back when they publish the data in public journals.
We got a few other.
You are mute, Mark.
Yeah, thanks. We got a few other questions. So going back to Stephen, I do want to say also to Cindy in response. Again, as a foundation, we have required those reports, and anything we participate in, we've actually, we push for publication. We give them a timeline that if they don't publish by a certain date, we do have access to that information to be able to share. That's been pushed back on by several researchers. We get it, but it's really important that sharing does occur. In addition to that, I will say that the researchers that we've funded and have dealt with, we have found them to be quite good at sharing. So when they have different conferences, they do get together, and they're you know, they're not egotistical fellows or people. They actually do get out and share a lot of their information and their learning. So it's been really nice to watch that. I'll go back to Stephen's comment. I don't know that samples from the early 70s are probably available now. I can let Stacy handle that one, as well as Enilton wrote, hello, I had a muscle biopsy in 2006 when I found out that I had NM. I could check up the clinic. I don't know, maybe if you guys wanted to reach out to Stacy, she could do a little bit of digging and see if she could you know, facilitate that process for you to be able to dig in to see what their storage capacity was like, and see if there was a way of sharing a portion, or if they were looking to, you know, transfer the entire process to the CMD-TR.
Yes, if anyone has questions about your specific muscle sample, just send me an email and we'll work through it together.
Mike, I don't know if you want to take Aaron's question. Does the age of the donor at the time of the sample was taken will make a difference in the variance of data collected, i.e. infancy versus childhood versus adulthood versus adult? Is a variety more helpful when doing research?
So I would say yes. I mean, it depends on the questions, but then the question, you know, your early questions, right? You're trying to get an answer on what to study. And so sometimes when you're looking at what to study, it can be useful to have a bunch of very similar samples of a very similar age to look at, so that you minimize the variability with age. But then one of the very early follow-up questions is what happens earlier, what happens later, what happens in boys, what happens in girls. And so in both, you know, in both cases, it's honestly good to get whatever's available. You know, just whatever happens to be, you have a sample for it, then, and it's possible to be donated. There are studies that will use it, that the questions should be, you know, good. And one of the things that we have the opportunity to do with the, you know, when groups say, hey, we want to study X, Y, or Z, is we can kind of, you know, we can kind of guide that a little bit too. They come to us with a bad experimental design, we can say, okay, we'll give you the tissue. This is a good question. Maybe change this around a little bit, and you're more likely to get a clear answer that, you know, and then do this in multiple stages, for instance, instead of all at once. So I think that, you know, overall, any sample from any age is great. I mean, even for like that Llama 2 project, there are samples within that project from the same patients, you know, a few of the same patients early on and later on. And that's going to allow us to address certain things about disease progression within a given patient within that study in a way that we wouldn't have been able to touch on at all without it.
Got a buzz going in the chat room, so let's try and go in time order. Tracy Cahoon wrote, we have some upcoming labs that are needed for my son. Can a sample be provided at the time for a cell repository?
Yes, absolutely. That is the most optimal time to be doing this. I advise patients that if they're having blood draws done anyway, that's the best time to do this. I can walk you through how to do all of this if you just want to send me an email, Tracy.
Oh, and then just, sorry, there was one above that about whether or not there's a way to do the default medical wristband thing. I don't know of a way to do that. I do think that, you know, there's some flexibility in terms of you don't necessarily need to know that you're donating tissue at the time of an emergency or whatever to donate the tissue. It's useful, for instance, to say, okay, there's somebody's going in for a specific surgery, can muscle be collected? But nobody that's doing an emergency surgery is also going to want to be messing around with research tissue collection, because they're going to be too worried about the emergency. And so our frequent types of tissue distributions from surgeries is either things for like a back surgery, for instance, a spine surgery. They do some cutting of muscle anyway for access. And then we'd say, hey, instead of discarding that tissue, can you bank it, you know, or can you freeze it and then send it to us when you're sure that's not needed? You know, and that's kind of a nice situation. The other situation would be when you have like a diagnostic muscle biopsy, like we've been talking about, and the clinical testing on it's complete. And so it's not going to be used for anything except for sitting in a freezer. And then you want to get that out of there before the hospital regulations might say that they're allowed to throw it away, which is usually a matter of a couple of years.
All right, Mike, we're going to pepper you a couple of times here.
Sure.
My daughter gets daily CoQ10, a supplement.
Okay.
So any pathological evidence shows the abnormal mitochondrial morphology in patient's tissue.
I submitted 2 papers on this yesterday, literally yesterday. So one of the things that we've been working on for a couple of years in our lab is whether or not mitochondrial dysfunction actually is a component of disease progression in Nemelin myopathy. It's not known, it's not necessarily agreed upon that it is. And so we've actually been working on it in animal models for a couple of years to look at what happens with mitochondria in Nemelin models. And the idea here is that, Nemelin's across 14 different genes, a bazillion different mutations and everything. And yet a therapeutic option here that would be broadly useful would be something that's not necessarily related to the primary gene, but something that's shared across genetic subtypes of Nemelin myopathy. And so in looking around at what went wrong in a number of these models, mitochondrial dysfunction is something that pops up. It's worse in the worst models. It's not present in the models that don't have any symptoms, things like that. And so, so there is a potential for, and the mitochondria are displaced by the Nemelin rods, they get tangled up and things like that. And so, there's the potential for a subset of Nemelin myopathy patients to actually benefit from things that manipulate mitochondrial biology. But right now we don't know who they are, right? We know that there's certain models that have mitochondrial problems, there's certain models that don't. And now the next stage of that is basically to use some of the human tissue to try to figure out which humans are like which models in an effort to identify, the kind of broad potential for manipulating mitochondrial biology. So I think it's an excellent question, and I'm glad that people are looking at your mitochondria, but that it's also something that needs a lot more work. This is kind of a very new idea, and these 2 papers that we submitted together yesterday represent a number of years of work that have been partially funded by AFBS that will really kind of put this idea on the map as a direction to go forward.
Some follow-up to that, Mike. Any EM imaging found any abnormal cellular components in patient's tissue?
Yeah, I mean, what happens in Nemelin is that you have protein aggregates, and then that displaces things, right? So when you do electron microscopy, which we do, you see the piles of Nemelin rods, and you see things that aren't Nemelin rods that are other piles of proteins. And then you see a bunch of displaced stuff. And so mitochondria are among the displaced things. And what we're kind of increasingly finding as we look harder and have better questions to look at is that the Nemelin rods have the things that people talk about, right? The thin filament proteins that relate to the mutations. But there's a bunch of other regulatory proteins and other things that are important for muscle to function that get stuck in them too. And so when you look at how much of a given regulatory protein is present, it may look normal or it may look high or something. And yet then when you look at where it is, they're stuck in these protein aggregates. And so that level of function that they're doing may actually be very low.
Exam, what's the most possible signal cascade associated with the fiber dysfunction in patient's tissue and the feasibility for treatment?
In terms of cascades, I would say the broadest involved cascade would be kind of the master mitochondrial regulators. So if you look at least the animal models, our look at the animal models that we had worked on a number of years ago and that we were just reporting really focused on broad spectrum protein expression, what's wrong, and then assigning that into categories and then looking at the categories that are wrong. And so you get your expected problem with muscle contraction, right? Because your primary mutation is related to one of the key contractile proteins in the thin filament for the vast majority of Nemelin patients. And so that's something to look at, but there's a number of different ways in which contraction is wrong. And that depends on the pathway or the specific mutation, the specific gene that's involved. And then for instance, ACTA1 is a thin filament component, but each of the different ACTA1 point mutations may do different things to actin that knock things off. And so, one of the reasons we've been focusing on mitochondrial function is that there seems to be a little bit less variation in that several of the key mitochondrial regulatory pathways seem to be abnormal in the symptomatic Nemelin myopathy mouse models. And that the idea of that we've been trying to get people interested in that work in these areas is, things that manipulate mitochondrial biology in a beneficial way may actually give a broad proportion of the Nemelin myopathy patient base some level of symptomatic improvement, even if we're not doing anything related to their primary mutation.
That's great. I want to approach a few things here real quickly. Sharon Smith asked, does a neurologist or geneticist order a muscle biopsy? I think it could come from either.
Either one.
Yeah,
Yep.
Either one. Sargon de Jesus, he wrote, are there ways to include the taking and transfer of tissue samples as a default for all medical procedures? For instance, I have a medical wristband that's connected to an online platform that doctors can access in the case of my incapacitation during an emergency, e.g. car accident. Is there anything I include in that profile to ensure that we don't lose a chance to gather tissue or even a postmortem sample?
So I tried to touch on this a couple of minutes ago, but I don't think I fully got through what I was thinking. So I got taken off track. So I don't know the answer to that. I think if you were able to, because it'll depend on what your band is capable of doing. I think if there's a way with your band to say that you're interested in an autopsy tissue collection, that would be, that would get the procedure done in the case of this emergency, right? Once it's done, then we can go and get it. Once we realize that something's happened to a Nemelin myopathy patient, and that somebody knows that this is an interest, in your family, the act of collecting the tissue is the time limited thing. But we've certainly had cases where, obviously it's a terrible time for the families, and they've expressed interest, but they didn't want to deal with all the paperwork and the consenting and everything, where we've just said, okay, well, we'll tell your institution how to do the autopsy. We'll follow up with you in like 3 months, and then we'll take care of this then, because we certainly don't need to, we don't need to be involved in this awful time in your life when you've got, so many other things going on.
And I would just like to add that it's really important to let your family members know that you are interested in tissue donation as part of your end of life planning. That way they can have the information. You can provide them with the name of our program, my contact information, so that if anything happens to you as an affected adult, your family members know that you want your tissue donated. They can then contact me if something did happen. And I would work with your local hospital for, like Dr. Lawler said, arranging for the autopsy to be done. And then we would arrange for the samples to be transferred to our biorepository afterward.
I want to acknowledge Aaron's comment again about letting families know that collecting samples at different ages is important, and that that the assumption that one single sample in a lifetime is enough. Actually, I think the rule, as I'm hearing it more today, is the more the better. So whenever a opportunity is there, we should take that opportunity. And Mike, is it true that even urine samples could be used to develop cell lines?
It is true. I think that would go. Stacy, does Coriel do that? I don't think we bank urine. It's possible to do it. I don't know that anybody banks urine for it that I know. And we're not set up to bank it appropriately. But we have participated in studies that, on the scientific side, I've participated in studies where iPSC cell lines have been generated from urine. I think if there was an interest in that sort of thing, we could certainly help find an appropriate group to help with that. And then well, one of the things is that we would want to make sure that they wouldn't just collect it, but they'd actually do something with it. So I know some people that would do it, that work in that area. I don't know their bandwidth to take in lots of samples that they weren't planning on. But certainly if there was an interest in that, I think we could help, help find a way to direct it that wasn't necessarily to us.
And just to clarify, the cell repository, they are looking for donations of blood or skin biopsies only, and those are transformed into cell colonies. And that's different from the tissue repository, where we take in tissue samples. We do not take in blood samples at all. We have taken skin biopsies in the past, but we're primarily interested in muscle samples and organ samples.
And then, similarly, if you have a question, certainly reach out to Stacy, because our goal is not necessarily to hit a certain number of samples within our bank. Our goal is to serve the community. And so if you have something that you want to donate, or you have an idea, it's okay. We can help try to connect, find and connect you with the right people to talk to about it. And it would still, it still should save you a lot of time and frustration to have us direct you in a certain way.
And related to that, I am contacted fairly often about donating affected embryos. We cannot accept affected embryos in our tissue repository, but there is a facility in Michigan that does accept affected embryos. So when patient families contact me about having a donation like that, I refer them to this other program.
Okay, a couple more questions quickly, because I know we're really over our time. If my neurologist recently ordered a genetic test through an in vitae blood sample, would that sample be available for your use, or would a new sample be required to be obtained?
Probably need a new sample, because I bet the blood sample, the blood sample will have had DNA extracted from it, that's going to kill the cells, basically. But sometimes there's a little bit of spare blood in a blood collection. Those aren't necessarily stored forever, and I don't remember offhand what the storage requirements are for Coriell to be able to use a sample and get cells from it. But throwing it in a freezer for just in case storage probably doesn't lead to viable cells on their end.
Right.
It could be worth checking.
Mm-hmm.
Okay, just on the side panel again, more chats. My son has had multiple surgeries over the years at the same hospital. Did you just keep in touch with them to have them keep sending you samples along the way for every time they opened up my child? Because if so, as a parent, I had no idea. If not, isn't that an opportunity to get your suppository filled? I think yes. I think the more we can communicate this effectively, and that's what the entire PCORE project is about, bringing understanding of research to the community, and then having the community again influence the research. And so we're sharing this information today is part of that goal. So I'm glad we're reaching out and we're getting connected to these participants here today. So I think this is a perfect forum. I apologize for missing out on past opportunity, but again, we're ready to move forward with more and more participation and filling that repository.
Yeah, and I mean that if you have multiple things, we won't know, I guess, is the key thing, right? So we won't know that you've had your son or daughter's had another procedure unless you tell us. The hospital won't tell us. It'll be a matter of if the consenting person says, hey, we had another surgery, we don't need another consent, but we won't know that it exists. And so even a simple communication saying, hey, we had another surgery, there's this sample there, feel free to go and get it. that would be all that you'd need to do. You wouldn't need to re-consent or anything. We just need to know what hospital it happened at. We could call them up, we could show them your old consent form, and then they could send it to us. But we just won't be aware of its existence without somebody connecting us to the fact that it happened. And so it'll never, a donation will never happen without your knowledge, because we have no ability to know that it exists.
And I think that's important to state. We don't have the opportunity to just go into hospitals and take tissue or to make arrangements with surgeons without the patient family permission. So the only time that a tissue collection would be occurring is if you had contacted me directly and stated, for example, my child is scheduled to have spinal fusion surgery next month, and I'd like to see if we can get a tissue donation out of this. At that point, I would contact the surgical team directly, explain our program to them, tell them that you are enrolled in our program and that you would like to find out if this is an opportunity for a tissue donation. Oftentimes it is. It's just a matter of the surgeon being willing to cooperate. So while the surgeon is in there doing what he or she needs to do anyway, like Dr. Lawler pointed out earlier, they are making incisions into the muscle. It's very simple for them to take a sample at that time. There is no added risk to the patient at all when it's done this way. So all they need to do is preserve it properly, and then they transfer it to us. And like I mentioned earlier, I do take care of all of those details. It's not anything that you need to think about. Your child is having surgery. You do not need any other things on your plate at that time. So all you need to do is tell me what the surgery is, when it's occurring, the date, and who the surgical team is, and I take it from there.
Great, thanks, Stacey. Mike, do you want to take that fiber type question?
Yeah. So you don't want to read the fiber type question? All right, fiber type question. So it's a massive type 1 fiber predominance, basically 95% type 1 fibers, 5% type 2 fibers. That's pretty common in Nemelin myopathy. It's not the usual situation, but there's certain subtypes of Nemelin myopathy where you get this dramatic fiber type skewing to oxidative or slow fibers, which is the type 1 fibers. And in general, in congenital myopathies, there's this kind of skewing in that direction. You're right that it does have bearing on treatment potentially, because there are treatments that may work better, when they become available for fast fibers or slow fibers. And so if you have a massive predominance of slow fibers in your muscle and you have something that affects slow fibers more as a treatment, efficacy thing, then you should expect more efficacy out of it, because you have a greater proportion of your fibers are actually going to be amenable to it. And conversely, if something's really good for fast fibers, you're going to have less efficacy from it. So this is one of the reasons that it's good to know, to have some of this information. It's good to have some of this information available in the samples that we have, so we can interrogate that stuff while treatments are being developed. But I've certainly seen treatments, especially for gene therapy, for instance, there are certain doses of gene therapy that are kind of in the middle of efficacy that sometimes segregate out to being more better biodistribution in slow or fast fibers. It flip-flops depending on what a bunch of other variables. But there is the potential for that to actually, impact how likely something is to be efficacious.
Again, I'm going to remind people that we can follow up with more questions at our next webinar on May 21st on Saturday at the same time, 1 p.m. Eastern. Thank you, Kate. I think there was maybe one more question. I see all good comments in the chat. I think it's all great stuff to share. We'll continue. We'll have 7 or 8 more of these webinars. And so we'll be able to kind of follow up and continue to share information that is vital to the success of finding treatments for Nemelin myopathy. So everyone, thank you so much for your participation, your involvement, your dedication to supporting those with NM. Really thank the team, especially Dr. Mike Lawler and Stacey, for covering a lot of the information today. Super helpful. Mike, we're going to let you get to your daily chores
Thank
earlier
you.
than you hoped. Thanks so much for everybody, and we'll be sharing the recording of this video to all the registrants probably in the next day or so.
Okay, well, thank you all for your attention, and have a good day.
Bye, everybody.
Thank you, everyone. Bye-bye.
All right, thank you, everyone.